Welcome back to online NPTEL course on Introduction to Complex Biological Systems. Now we are in module 6, where we are going to discuss about cell, tissue and organ, particularly the complex architecture of living system. So in module 5, Professor they discuss about cell, the smallest unit of life. But here we will go into more complex topics, that how multicellular organism developed. So let's start. So today particularly I will be discussing about prerequisites for the development of a multicellular organism and then establishment of different cell types. As you know that in multicellular organism, for example in human, we have different types of cells in our body. So how those cells are getting generated, mostly I will introduce all those things here. So here I am showing the cycle of life of a multicellular organism. So as you can see, if I start from here, like this is zygote. So just after fertilization of sperm and ovum, it forms zygote. So if I am talking about human system, then here we have all the chromosome that should be present. So in this case 23 pairs of chromosome, right? 23 pairs of chromosomes present in zygote. And now it will undergo mitosis and it will form morula. So this stage. So now as you can see here only few cells are present, and these cells they have the property, they are called stem cells. You already know from your module 5 lectures. So now these stem cells they have the potential to make different types of cell in our body. So as a result of that, it will undergo again mitosis and it will differentiate into different types of cell types, as well as it will form some organ, and finally we will have the embryo. So this is the early developmental stages. All these things are happening because of the mitosis division. As you already know that two major types of cell division, mitosis and meiosis. So as you can see, all these processes are happening because of the mitosis. Now again from embryo, mitosis will be going on and morphogenesis will be going on, and finally after some time we will have adult individual. As you can see, this is, we are just putting example of human here. So as you know that in adult, like approximately 10 to the power 13 cells present in human. So now those cells are being produced because of the mitosis, and as you can see in a complex body plan, we have specialized organ, everything is in proper place, and that will help us. So now in adult, now meiosis is also going on, and because of meiosis some specialized cell, germ cells, they will form sperm in case of male individual and ovum in case of female individual, and finally after fertilization again we will have the zygote. So in ovum, in that case we have half of the chromosome, that means just 23 chromosome, not 23 pairs, just 23 chromosome. Similarly here 23 chromosomes in sperm. So we say this is n number and this is n, and finally in zygote we have 2n number of chromosomes. So this is kind of cycle of life in multicellular organism, because in case of unicellular, for example bacteria, the system is completely different, right? So from one bacteria just their chromosome will be replicated and then the cell will divide into two, but they will not stay together. those two cells will be different. They will be different individual. So because they are unicellular organism. But in case of multicellular organism like plant, animal, including human, we have lot of complex organization and complex processes, and I am going to discuss few of those things today. So these are the essential processes for the development of a multicellular organism. As you know, one important thing is cell proliferation here. So that produces many cells from one cell. As I already mentioned that, for example, every individual like human, for example, we started from just one cell, from just a zygote. And adult individual, I just told that approximately 10 to the power 13 cells present. So as a result of that, you can understand that cell proliferation is very important thing that will produce many cells from just one cell. And now here the next thing is cell specialization. So this is creating cells with different characteristics at different position. For example, as you know that our nerve cells, they have their unique features and they have unique function. On the other hand, some cells present in liver or pancreas, they are also busy to do their specialized function. So cells need to be specialized over the time during development. That is also an essential step during the development of multicellular organism. And very importantly, number 3 is cell-cell interaction, or just say cell interaction. Coordinate the behavior of each cell with that of its neighbor. The thing is, although we have many cells in our body, but they are kind of regulated. They coordinate each other, they influence each other so that they can help different types of specific function in our body. So this is absolutely important for a multicellular organism. And then cell movement. As you can see here, we are showing that 4 different types of cell. As you can see, this one, like we are just marking with different color, but you can see here that those cells are moving in a specific pattern so that they can rearrange to form different types of structured tissue and organ during the developmental processes. So this cell movement, this is kind of, I would say, like very unique features in case of animal development. As you can see, in case of plant, like in plant development, this cell movement is almost, cell movement is not there, because as you know that in case of plant, those cells, they have cell wall, that is very hard structure, and those cell walls kind of, they are cementing those cells in the surrounding region. They cannot freely move here and there. So as a result of that, this is a major difference like the cell movement in case of animal development as well as plant development. So these are the 4 important prerequisites for the development of a multicellular organism. Here I am just introducing that the developmental defects arise from misexpression of genes. Whatever 4 steps I mentioned there in the previous slide, if some dysregulation happens, some problem happens because of some, maybe it can be because of some genetic level problem, then it will give some developmental defects. So here we are showing one fly, Drosophila. So as you can see, this is the eye of Drosophila. And now there are many genes there involved for the development of eye in Drosophila. One of the important gene, here I am saying that important protein, that eyeless protein, which is also known as PAC6, this is product of some gene. So that controls eye development in Drosophila. But when it get misexpressed, its expression is not controlled properly, is misexpressed, then we can see the development of eye in the abdominal side. For example, as you can see that here eye-like structure, you can see that eye here, where this is just wing, right? So, misexpression of eyeless or PAC6 in wing precursor produces eye in a different location, which is not the normal location. Similarly, here you can see eye, development of eye on leg of this Drosophila. This is again because of the misexpression of this protein. Now the question is not just misexpression of this eyeless protein in Drosophila. Like scientists observe that the homologue of this protein, I would say the same, or I would say the similar gene which is responsible for the development of eye in other animals, and if that is misexpressed in transgenic fly, this will also result similar scenario. So therefore, this kind of thing is very much conserved also. The expression should be strictly regulated. The misexpression will result into developmental defects. But fortunately, in most of the time, everything is going properly in our body. That's why we are healthy and happy. Now, I will start about the overview of development. Here, conserved mechanism that establish the basic body plan. So particularly here, whatever I am trying to explain, this is related to development of animal. So as you can see, this is fertilized egg, or we can say this is same thing, right? Zygote, fertilized egg. And after cleavage or several round of initial division, it will form blastula. And these cells present here, this is called blastomere. These are kind of epithelial cells and they are blastomere. And now it will undergo another process called gastrulation. This is complex process, and during this gastrulation process, as you can see, we are showing cells with different color in here. This is gastrula. So now you can see whatever cells present here in blastula, after this stage, after gastrulation, some of those cells tuck inside. So as you can see, this green cell, they are tucked inside. So these green cells are called here in this diagram, they are mesoderm. As already mentioned in this figure, this is mesoderm. While we are showing these cells on the outside in blue color, they are epiderm, or I would say ectoderm here, ectoderm. And cells which are going inside, shown in yellow color here, that is endoderm. So as a result of that, what I am trying to explain, during gastrulation, some of the cells stuck inside and they are forming three different layers. On the periphery, we have ectoderm, and inside we have endoderm, and mesoderm is kind of sandwiched between endoderm and ectoderm. And more importantly, I would like to mention here that ectoderm here, this is the precursor of epidermis, for example, skin layer, right? This is the precursor of skin, as well as whole nervous system, they are also produced from ectoderm. And this overall idea, this is true for for every animal, I would say, in human, in frog. So this is mostly whatever we are trying to explain. This is based on experiment on frog, but this is kind of conserved body plan. And now the endoderm, whatever I told at the inside, at the core, this is the precursor of gut and its appendages such as lung, liver, pancreas, all those organs, those are formed from this endoderm. And the mesoderm, which is sandwiched between endoderm and ectoderm. This is called mesoderm. This is the precursor of muscles and connective tissues. Connective tissues, for example, our blood cells, blood vessels, all are produced from mesoderm. So as a result of that, you can understand that at the very early stage of development, those things are getting decided from this position and patterning of ectoderm, mesoderm and endoderm, which will form different complex system and organ when the development is going on. So here, as you can see, the conserved mechanism established the basic animal body plan. Whatever I was discussing here, we are showing an early stage of embryo, and this is again, this is example taken from some frog embryo. So, and now here in this left side, as you can see here, we are like trying to explain different plane. I would say here the dorsal side and ventral side. So if I see here, like I'm trying to show here, so this is dorsal side and this is ventral side, right? This is ventral and dorsal. And then we have posterior, this is posterior and this is anterior, the front part and the back part. And then we have right and left, this is right and this is left. So this way, for our discussion, we just we are just mentioning in this way, but this anterior posterior determination, dorsal ventral determination, this is very important for the development, otherwise everything will be skewed up. So as you can see, if we like just make a cross section through this embryo, as you can see on the outer surface we have the layer which is shown in blue color, that is the ectoderm that form the epidermis. And as I already mentioned, that ectoderm is also playing important role for the formation of nervous system. The neural tube formation takes place from the ectoderm itself. And now at the very middle, here we have the endoderm and yolk. So this shown in yellow color, endoderm, and inside the endoderm you can see some hollow cavity like structure here. We are saying that gut cavity here, which is lined by endoderm. And now mesoderm is just a layer in between endoderm and ectoderm, as we already discussed, and this part is the head and this is tail bud. So as a result of that, after a certain time of development, it will be very much clear that this is head and this is tail, and then other organ will form progressively. Now whatever I told, that was kind of little bit of introduction. If I take a specific example here, that how these different types of cell or organ develop. In this case, whatever I mentioned, this is because of at the beginning, like for example zygote, it has like huge potential. It is kind of stem cell. So zygote can divide into multiple cells in that those are called blastomere, like, but they are stem cells. They have the potential to make different types of cells in our body. And after gastrulation, when we have epidermis or the I would say the ectoderm, endoderm and mesoderm, now they have some kind of restricted potential, that mesoderm will form blood tissue or those kind of tissue, connective tissue, but mesoderm will not form nerve cell, for example, because that is already restricted. So this is the point here, that the developmental potential of cells become progressively restricted over the time during development. So here blastomere, this is, as you can see, one of the many cells formed by the cleavage of a fertilized egg with a potential to give rise to most or all cell types, as I already mentioned. Now in the next step, when it is forming endoderm cell, then it has some limited option. It cannot make everything. It has potential to make make the gut and some other appendages in our body such as lung, liver, pancreas. So still it has potential to make different types of organ in our body during this stage. Now if I go little bit more, for example, in the next stage, as you can see, this endoderm cells is forming now the next cell types, that is pancreatic bud cell. Now this is kind of specialized cell that will give rise to other pancreatic cells. So that means this is now confined to pancreas only. It will not be able to make lung, liver or gut. This is kind of already going into the pancreatic line. Now again, after some division, it can make endocrine pancreas cells. So in pancreas there are many types of cells. Some of those cells are endocrine cells. They are producing hormone for us, right? For example, islet cells, also called as islets of Langerhans cells, produce and release hormones. For example, insulin, glucagon, those are the hormones. They are very essential for our body, and those are produced from islets of Langerhans. So as a result of that, when this blastomere through these processes, it reaches into this stage like endocrine pancreas cells, that means their fate is to produce hormones, right, inside the pancreas. And now they can again be specialized and they can form the beta cells in the islets of Langerhans, beta cells in the islets of Langerhans. As you know that beta cells in the islets of Langerhans, they produces insulin for us, right? So now they are not even able to produce glucagon, the other hormone. So they are very much specialized and they are forming and storing insulin for us. So this way, progressively, stem cells can be converted into more and more specialized cells, and with like specialized cells with specific function, as we explained in case of this pancreatic beta cells which produce insulin for us. Now, why these things are happening? Because we must consider that all those cells, even during the gastrulation process, I told that three major different types of cells, right? Ectoderm, endoderm and mesoderm. But all those cells, they have same set of information. They have same DNA, right? But still they are behaving differently. They are going into different, different options, and they are making specialized cells and organs in our body, right? So because each cell type expresses a unique subset of genome, and that product means, I always mention product means here RNA and proteins, give the cell types its unique function. And because of that, the control of gene expression is very, very important, because although we have the same information in every cell, but how those genes are getting expressed, that is very crucial for this different type of cell fate. And there is a combinatorial code of gene expression for each cell type. I mentioned in some during some different discussion that we have around 20,000 different protein coding genes, but they have very complex combinatorial code, that some genes are constantly expressed in all cells in our body. Some genes, they are not getting expressed at all in some cells and tissues. So as a result of that, it's a very complex process, and that decide the production of different types of cells in our body. Now here very simply I am going to discuss that what is this means, the combinatorial code of gene expression. So here if you see, I am trying to explain in three different types of cell. One is nerve cell, and the next one is muscle cell and skin cell. So those three cells, they have different location in our body. they have different function, and they are completely differently specialized for their function. But if you see, their genome is same. In their genome they have whatever I told, 20,000 protein coding genes, all are present. But here for the explanation, I am just mentioning only here, just for explanation, that 5 genes A, B, C, D, E present in all these cells: nerve cell, muscle cell and skin cell. Now, out of these 5 genes, as you can see that gene A here present and it is expressed. So, here we are showing gene expressed. So, A is expressed in nerve cell, muscle cell, skin cell. We are showing just 3 different cell types. Maybe if we include more cells like liver cells and pancreas cells, we will be seeing that A is getting expressed in everywhere. So, as a result of that, I would say that gene A is housekeeping gene that is expressed everywhere. Now, the next one, gene B and C, they have restricted expression. So, as you can see, B is present or B is getting expressed in nerve cell and in skin cell, right? Nerve cell and in skin cell. So, both are originated from ectoderm, right? But B is absent here in muscle cell. Muscle cell is produced from mesoderm. So, as you can see, they have some kind of restricted expression. So, that is why this B and C, they have restricted expression. And if you see D and E, they are cell type specific. As you can see, E is only getting expressed in nerve cell, and that is giving unique features to nerve cell, while D is only getting expressed in muscle cells and providing unique features to muscle cell. So, this cell type specific genes, although we are saying just E in nerve cell, but we might have many other genes also, they are specific to nerve cell. But this is very simplistic view of this combinatorial code of gene expression, and therefore you can understand how it can regulate or it can determine different types of cells with their specific function. Now, this differential gene expression largely responsible for the differences between different cell types, as I already mentioned. But why these different genes getting expressed in different types of cell? Although the information is present everywhere, all this is happening because of the regulatory modules and the regulation of gene expression. As you can see, I am showing here this DNA, and in this DNA I am just concentrating 3 genes here, gene 1, gene 2 and gene 3, and those 3 genes are actually present in both muscle cell and skin cell. As I already mentioned, the information is same everywhere. Now, at certain point of time during developmental time, as you can see, in muscle cell, somehow the gene 1, this is our gene 1, right, shown in here, gene 1. So, gene 1 is not making any product because of some kind of regulation. On the other hand, you can see that gene 3 here, gene 3 in skin cell, it is not also making any product. So, therefore, as you can see that only gene 2 getting expressed in both cases in skin cell as well as in muscle cell, but gene 1 and 3, they are kind of differentially expressed in 2 different types of cells, muscle cell and skin cell. This is because of the availability of these transcription regulators, some other proteins, they are controlling the expression of this gene, and that actually determine the fate of these cells, what kind of product they will make, and accordingly they will make different types of cells in our body. So also through combinatorial control and cell memory, sometimes simple signals can generate complex pattern. As you can see, like instead of specifying that particular gene 1, gene 2, gene 3, whatever I discussed before, here I am trying to explain in a defined way. So as you can see, these two cells, like here, cell number 1 and cell number 2, they have same properties. That's why we are showing kind of same color, same shape, everything. So they are kind of same cell, I would say. Now, the combinatorial control means when these cells are getting, divided, or when these cells are undergoing, during the developmental processes. So as you can see, in cell 1 here, we have some signal A and signal B. They are kind of influencing the fate of this cell 1. If I say this is cell 1, this signal A and signal B is influencing the fate of this cell 1. While in case of cell 2, here we have the signal A same, but signal C is different, some other signal. Signal means here it can be some, some protein, some hormone, some any other, chemical like found in our body, that can be some kind of signal. So this signal A and signal C, it is also influencing cell 2. So as a result of that, now these two cells, you can see they are different, like they are different now. So here these two cells, they are getting different. Their property will be little different also. So as a result of that, this is kind of, I'm trying to explain you the combinatorial control. And on the other hand, here this is even more complex, that control at the level of cell memory. So that means here again, if I mention this is cell 1 and this is cell 2, and when we are providing two different types of signal, I would say some kind of two different types of influences, like signal X and signal Y. So as a result of that, those two signal actually kind of alter those two cells. They can remember, those two cells can remember those, they receive two different types of signal. And that's why I just, change their color, the nucleus color here, we change here, as you can see here. So this is different signal, somehow they can remember. On top of that, when they are getting the same signal, signal C, by these two cells, again you can see two different types of cells are getting generated. So here, what is the difference between this left side and right side? So here at the end, signal C is same in both cases, but still, because those cells previously they received two different signals, signal X and Y, and they are staying as memory in those cells. So as a result of that, upon receiving signal C, they are giving you some different types of cells in our body. And apart from that, I would also like to mention that small number of conserved cell signaling pathways coordinate some kind of patterning in our body. This is very important. Although we are very different, for example, one Drosophila, one worm, or I would say one bird, human, we look very much different, but we have some a few conserved cell signaling pathways. For example, I would say TGF beta, notch signaling. So these are very common signaling pathway. Those are conserved in many animals. So now, what is this inductive signal? As you can see, these cells here, we are just putting cells one after one here. So now these cells at the center, they might produce some kind of signal. For example, this TGF beta. TGF beta stands for transforming growth factor beta. So this TGF beta will induce surrounding cells to be developed into a different types of behavior, different types of cell. As you can see, because of this signaling effect, these cells are now getting changed from the surrounding cells. So as a result of that, these cells directed to new developmental pathway because of these inductive signals. And this signal can travel through different methods. For example, when cells are close by, cells are interacting with each other, right? So two cells, they are getting attached by some kind of cell-cell junction. So as a result of that, nearby cells, they will receive these signals. Also, this is also possible that some of these signaling molecules, they can travel, they can diffuse over a long distance, and they can also modulate the function of those particular cells. Then how it will happen? Because maybe the receptor of those particular signaling molecules present on those cells which are receiving, although they are present little bit away from the source, but they have the receptor which will bind to this signal. For example, TGF beta. Like TGF beta will bind to its receptor present on some other cells. It might be little away, but still it will give rise to, like, it will help to change the fate of those cells because of this signal. Here, the morphogens are particularly long-range inductive signals that exert graded effect, as you can see. So we are showing in 2 dimension only. As you can see, those cells are just present in this way, but like although we are showing here, but you can also understand in 3 dimension that everywhere cells are present, and maybe this is the source cells which are producing some kind of morphogen. So it will be diffused from the source like this. So as a result of that, as you can see, if it is diffusion, then the nearby cells will have more concentration of this morphogen, but here we will have intermediate level of morphogen, and here very less, right? Very less amount of morphogen, and this is intermediate, and this is high level of morphogen. So finally, that will make those cells different, and they will have some kind of specialized property in those cells. So as a result of that, this morphogen also help to determine the specific function of specific cells present in the surrounding. And here this is the last concept for the generation of different types of cell here. So as you can see, the asymmetric cell division can also generate different type of cells. So here this is stem cells, right? So this is stem cell, this is stem cell. So as I already mentioned that in blastula, those blastomere, they are the stem cells, right? They have potential to make different types of cell. But also I would like to mention that in adult individual also we have some stem cells. For example, in our bone marrow we have stem cells, right? So from there constantly we are getting different types of blood cells. So as a result of that, although during the early developmental stage we have huge amount of stem cells and they have like huge potential to make different types of cells, but in adult also we preserve some stem cells for a specific function. Now, as we can see, this stem cell, when it get divided, so if stem cell getting divided and they are not forming stem cell again, then we will not have the stem cell when we will grow. And I just mentioned, even in adult individual we have stem cell. So as a result of that, when stem cell is getting divided, those stem cells somehow they have maintained some kind of lineage, some stem cell they need to preserve, right? That is happening because of the asymmetric cell division. As you can see, when these stem cells getting divided, it will make one of these cells called intermediate progenitor cells, IP here, intermediate progenitor cells, and one stem cell again. This intermediate progenitor cells, it will divide again, and that division can be symmetric division, and because of the symmetric division it will make some identical cells, as you can see here. That is because of the symmetric division. And now these, after symmetric division, these cells, because of like whatever I mentioned, that because of differential gene expression, some inductive signals, and so many things that can again alter the fate of these cells. But somehow this stem cell, they are maintaining this stem cell line. Again, when this stem cell will divide, again it will make intermediate progenitor cells and it will keep some stem cell line. So this is the beauty of this asymmetric cell division. So therefore they can maintain the stem cell lineage and they can also produce differentiated cells here. So now this is very important for the development of different cell types. As you can see that you might get a different types of cells here from this IP. Similarly here you can get a different types of cell from this IP or intermediate progenitor cells. Now, what is the fundamental basis of this asymmetric division? Because when the division is happening, for example, this is, I am showing asymmetric division. You have to be very careful here. Although this asymmetric division is happening here, but the genome, the information is same in both the cells here, this cell and this cell. They have the same set of information, same genomic content. Genome wise they are not different, but somehow still they are making different types of cell, different types of fate in our body. That is because during asymmetric division what happened, like some of those important product, I am not going into the details during this lecture. In the next lecture I will discuss in more details. So here, for example, this might be specific RNA, it can be particularly mRNA, or it can be specific type of proteins. They are somehow getting segregated in one pole of the cell, on the one side of the cell. So as a result of that, after division, as you can see here, these cells now they are receiving those special molecule, whatever those are mRNA or protein, that doesn't matter here, but they are getting those special product, and that will influence this particular cell into make some different types of cell compared to the cells I am showing on the top. But they are not receiving those special molecule which are the determining factor here. So this is asymmetric cell division, and because of that, from the same cell you are getting two different types of cell, and gradually they will make completely different type of cell types, right? And another thing here is very important, that sometimes symmetric division, although like I just mentioned about the asymmetric division, but sometimes symmetric division, as you can see here, this is here symmetric division is going on here. Two cells are same, but after division, this cell at the bottom I am showing here, this one is getting some extrinsic signal from the surrounding. As a result of that, it will give rise to a different type of cell compared to the top cell here. So this is another way that two cells are getting different. This extrinsic signal, it can be some kind of whatever I told that, some inductive signaling or some other kind of signals, and that also result into two different types of cells. So all those things, different types of gene expression, combinatorial gene expression, as well as asymmetric cell division, all sort of things actually determine or that decide the fate of different types of cell types. They produce different types of cell types, and from there, as we progress during the development, it will make complex organ in our body, and this is very important for the development of multicellular organism. Okay, so this is all for today, and mostly for this part you can refer Molecular Biology of the Cell by Alberts et al. And like many of those figures are actually taken from this textbook, and that's all. Thank you very much.